I'm Nick Andrews, Product Manager at Thermo Fisher Scientific, part of the Human Identification Team, or HID as it's known. Today, we're here in sunny Pleasanton, California, the home of the HID Research and Development Team. We have something super exciting to share with you today. We are ready to unveil the latest and greatest genetic analyzer, the SeqStudio Flex. Now, you may know that the SeqStudio Flex has been available for the research community, but we're ready for forensic prime time. So today, we have 3 forensic experts from the Sacramento County Crime Lab who are here to put it through its paces. They'll get to see all the greatest features, like the 4-plate deck with continuous plate loading, sample reprioritization, the integrated data collection software, and the remote monitoring and remote troubleshooting features. I know this instrument is going to blow them away, and it's going to blow you away too. So let's go meet the team. I cannot wait to see what they think.
My name is Ryan Nickel, and I work for the Sacramento County Crime Lab. I'm a criminalist there, and I'm also a crime scene lead at the laboratory.
I'm Natalie Jones. I also work at the Sacramento County Crime Lab as a criminalist, and I'm also the local CODIS administrator.
I'm Danielle Frost. I'm also a criminalist at the Sacramento County Crime Lab, and I'm in training.
Well, thank you so much for introducing yourselves, and thank you very much for coming in. This is great. You all seem to have a lot of different experiences, so this is going to be a really exciting time. Just to confirm, you're all forensic scientists, none of you are paid actors.
Not an actress.
Right.
So what do you actually know about the SeqStudio Flex? It's been out for the research community for a while, so have you heard anything about it?
Nothing.
Only the name, which I saw in the email that we got. And
CE instrument.
Yep, that's right. It's our brand new latest and greatest genetic analyzer that we have. It got released for our research community a while ago, and now we're ready to unveil it for the HID market. So we want you guys to be the first look at this instrument. So
we're the first forensic scientists to do this?
you're going to be the first forensic scientist in the world to touch this instrument.
Wow.
That's exciting.
That is
It's going
exciting.
to be exciting.
It's cool.
It's going to be great. So if you had a new CE instrument for your lab, what would you want it to do? Would there be anything you'd be looking for?
I would probably something along the lines of, if one plate is running, if you could actually load a plate on while the other plate is running would be great. That would be a big help in our laboratory, for sure.
Good. Glad you say that.
I think it would be nice to be able to load more than 2 plates. 4, maybe 6. That way we can get like everyone at the lab working on at the same time and just turn the instrument on and let it go.
I would love for the software to be really user-friendly and to be able to easily load on your plate and press button and say go.
Right. And you mentioned that you were currently in training.
Yes.
So how has it been learning how to use CE in the application of forensic DNA?
It's been challenging just learning, memorizing the steps. There's so many steps that come with the CE in like managing the software. So it's, a question of where do I look for this button? It's kind of hidden away in this corner. And so that's been difficult. So.
Right, yes. I know from my time in the lab. And you all have 3500s in your lab, correct?
Yes.
Yeah. So what do you like using about the 3500, or how has it helped you?
I like the anode and cathode buffer, that it's just, you don't have to load it or make any mixture. It comes in a pre-made box, can throw that right on. I also like the, it scans the polymer for us and tells us how many injections. So that's nice. I like that we have, our lab uses 24 capillaries, so we can get through a lot of samples pretty quick.
Yeah, we use USBs, so we have to go into the run room, load it onto the USB, take it back to our desk, load it into the software. So I hadn't thought about that, but that would be nice if we could like remote into that computer from our desk, pull our data, put it right into the software.
I would love that.
Yeah.
I'm sure actually other analysts that are, we're allowed to work from home a couple days a week, would actually like to remote in, get their data, and actually analyze data from home too.
If you need some re-injections, you don't have to worry about replating when you come in the next day.
Yes.
That'd be cool.
Yes. Can this instrument tell us at our desk that something's wrong? And we can pop in there and fix it.
Well, I don't want to give too much away, but it might have some of these features that you've been looking for.
I like it.
I'm pretty happy that you brought these up, but I think maybe we should probably get in there and actually see this instrument.
All right.
Yeah. Let's see it.
Cool.
Let's do it. Okay, well, Ryan, Natalie, Danielle, Eager Beaver, I would very much like to introduce to you the new SeqStudio Flex.
Wow. Wow. Very sleek.
Yes. Ooh, that is cool.
Now can I touch it?
Yeah. How can we touch it?
Go ahead. I had my fun. Now you can get in there.
Like looks very similar back there.
Yeah. Can we take a closer look?
Please.
I'm assuming touch screen.
I want to know how many plates. Can I touch the screen?
Can we open it?
Yeah.
Boom. 4 plates, guys.
4.
You can see the drawer is unlocked. Oh.
Go ahead.
Oh, they're not there. They're there.
Yep.
That's fun.
Oh.
So you can load while it's running.
Yes.
Wow.
Continuous plate loading, sample reprioritization. So you can load a plate. It could be injecting. You could then load another plate. You could say my injection's more important. You could put that to the top of the list and it will take that plate. You could have 4 plates in there. When 1 plate is finished, you could open that drawer, take it off, and there's a new spot open again. You could maybe wait until the end of the day, load 4 plates at once, and it can just run all night, and then you can come back into data in the morning.
Wow.
I know one of your concerns might have been about, well, what happens if there's an injection failure or something like that? But this also has remote monitoring. So you can get alerts at your computer, you can get them on your phone, you can even do re-injections from your phone with this instrument. So if you are working remotely, you can do that.
Yes.
And for those of you who maybe used a 3130 back in the day where you had the barn doors, this instrument has the same type from there.
Very cool.
Easy to get in there. Obviously your oven door. You may not be able to tell, newly designed array as well, so it's actually easier to install than even the 3500.
I love that.
It looks easier too. Like you can kind of see just where the little buttons are there.
Pop in, out.
Exactly.
So can I open the drawer right now to the instrument?
Yeah, as long as that screen says the drawer is unlocked, you can open that drawer at any point. Okay. Go ahead.
My question was, is there like an arm that kind of grabs those plates and takes it up to be analyzed?
That's exactly right. So the arm will pick up the plate and move it up to where the capillaries are, so you can actually do the injection itself. So that drawer is unlocked at any point during the run, so you can load plates. The only time it would be locked is when an injection is in place. Just for safety reasons, you've got high voltage running through there. Right. But otherwise, you can load plates on there. That screen will also tell you when you have plates that are present on there, so you can see what is linked, and you know if there's available space on it.
Nice. And do the plates set in or do they clip in?
They just sit on top of there. So as long as you put them in that plate cradle, then they just sit in there and they wait for the arm to come and pick them up.
Very cool.
What about the runtime?
It's actually about the same as the 3500, so it's kind of around the same time that you had before. A couple other features while we have the drawer open, there's also an integrated barcode scanner in there. So if you use barcoded plates, it can read that so that you actually know what you're linking and you can verify it. As before, you just had to make sure that you put the plate in the right place. Yeah. This time you don't.
I see 2 USB ports right there. Is that to like bring in our data and it automatically kind of populates into the screen there?
So we knew that there's obviously a lot of different options that you can have when you want to transfer data. This instrument does have cloud connectivity and local area network as well. So you can network the instrument on your local area network in the lab. You can set up your plates from your office and you can have those plates loaded directly onto the instrument through a piece of software called Plate Manager, which we'll show you in a bit. And then you could also do a USB. So you could put it into like your standard CSV or text file, load it onto the instrument, or you could do it through the cloud. So you could use Plate Manager on the cloud, set up your plate, and it will remotely send that plate to the instrument itself.
That's cool.
A bunch of different options. One thing I did want to do is introduce you to 2 of our R&D scientists who've been working really hard on the validation of this instrument. They are going to help us set up a run and get the instrument going and teach you how to use the instrument thoroughly. So what do you say we go meet them?
Yeah,
let's do it. All right.
Hi, everybody. My name is Nancy Liu. I'm a staff scientist at Thermo Fisher. I've been working on this project for over 1 year. So today I'm going to show you how to use the Plate Manager. That's a software used for creating your files for running the samples on SeqStudio Flex. So we have 2 different versions. So one version is on the cloud, another version is on the desktop. So this is the version on cloud. So you can create your file from the scratch by clicking on this guy here, or you can open a file which has been saved on cloud or from your computer. Or if you want to use a recently used file, you can click on this one. You can check, see whether it's correct or not. If everything is correct, so you can send this to the machine directly. You can choose multiple instruments at the same time and then click on OK. So then this file would be sent to your instruments. Or you can just choose one of them. So like today, I'm going to just send this one to this instrument. So after you select it, you just click on OK. So now the file is on your instrument. You are ready to run your sample on the instrument. And also, I'm going to show you how to create a file using Play Manager. I will be using the desktop version today. So I'm going to show you how to create a plate file from the scratch. So to do that, you just click on this one here. Then you get all of these things, by default. And you can change your name here. You can change the name of the file, or you can just use the default file name. If you say, OK, all the parameters here are correct, so you click on Next. Then you can click on the quadrant you want to put your samples in. And then you click on here. From here, you choose the injection protocol. So today, I'm going to show you how to choose this one. That's for the J6 dye. So after you click on that, see all the other parameters show up here. After that, you click on here, so then you can put the name of the files in there. So what I do, I just I have prepared everything, so I just copy the thing here, and then paste it to the software. Sample name, I click on this one and Control V, so then the file names are pasted here. And also, you can put the panel names here. So these are the panel names. All of them here, I use GlobeFinder, IQC, panel. So click on the software here. So then you get to this panel. OK, so you click on the first one. You click on Control V here, so in the file, these are the panel names. And then I put the kit names here. Highlight everything here, and then Control C, and put it into the first custom field. Then Control V. OK, after that, you go here, you save it, and then it's saved. After it's saved, you can send it to the instrument, or you can export it into a USB drive. So I can export it to either my computer or a USB drive. So save. Now, it's saved in this USB drive, and then I'm ready to use it.
Is the computer attached or connected to the instrument at all?
Yeah, so this computer is connected to the instrument through Wi-Fi.
Gotcha.
Yeah.
Nice.
And at our lab, we use an Excel program with our sample names and information. Yeah. So we would be able to export that in like a CSV file and then load it into the software rather than manually setting up our plate. Yeah,
so you can do that. So basically, after you set up the plate using Plate Manager, or you have a CSV file, installed in the on your computer or USB drive.
So instead of a computer, can you use your phone to do this?
Yeah, you can use your smartphone.
Okay.
Do the same thing. You can monitor your run And also you can unlink or link your plate. If you found that, after you go home, or if you are, somewhere else, far away, if you just, thought, oh, I made a mistake. So you can, make the revision of your protocol using your cell phone or your computer remotely.
Nice.
That's really cool.
Good feature. Mhm.
Yeah, as long as the plate is still, in the same location. Yeah.
Right.
You don't even need to come here to, unlink the plate. You can just use that. Do that with your.
Or set my injection before his.
Right.
Okay.
I also really loved that each injection was a different color. That was very pleasing to the eye.
Yeah, exactly. Me too.
So if there's somebody at your lab, like that always thinks their case is the most important and wants to jump in front of everybody all the time, is there a way to prevent plates from getting bumped to the front?
I think that would be a very good feature. But all these things should be tracked in the SAE feature.
So we could know who jumped in front
yeah.
of my run. Got it.
Exactly.
Hi, my name is Jimmy Lee. I am a staff engineer at Thermo Fisher. I'll be showing you the SeqStudio Flex. So this is their new instrument. So I'll have Ryan come and play with the instrument if you want to.
Sure.
All right. So if you want to load, I know Leonson helps you set up a plate, right? Yeah. So here, I'm going to hand you that plate. Okay. You can open the drawer. So there's positions A, B, C, and D. Already 2 have been filled, so you can put it in C or D.
Okay. I'm going to go with D. Oh, it already.
That was easy.
Slide it in. There you go. It will lock once you close it, but now you can link your plate, which has already been loaded on by Leonson. So click link plate. Yep. Go to My Instrument. And it's right there, the Demo 8 cap.
Okay.
And then you can check all of the properties. So you click on Application.
Click on Application. Oh, there it is. Okay.
See, it's already on HID, 96 roll plate, 36 centimeter pop 4. Click Done. Go to the plate screen. And here we're going to do an injection from column 2.
Okay. Yeah.
If you want to see what it is, you can go and highlight it. And those are your parameters. And you can click on the pencil button. And then you can scroll and see all the configurations.
Nice.
Size standard. Okay.
For the most part, you have your J6 sample in there.
Yep.
You click Done. And when you're ready to run, click Start.
Start run.
Yeah.
Is there a way to check the names of the samples that are in there?
You go back to that pencil. You can go to Sample Name, click on Multiple. And here's where you can enter all of your names in.
Oh, gotcha.
Johnson's already pre-named them here.
Right. There's the 8.
And you go to Sample Type.
Oh, and change the.
You can go there, and then the first one's a little glider, the rest are sample.
Gotcha. Okay. Cool.
Yeah.
Done. And then
Click Start.
Start run.
There you go.
Right on.
It'll do some pre-checks to make sure the consumables are not expired, and it'll give you an estimated end time, and there it goes.
Nice.
That starts up really fast.
Yeah, it did. Did it have to preheat?
So if you didn't preheat your oven, then it will have to preheat. You can actually do that before you run. If you hit that speedometer symbol on the top,
Yep.
if you scroll to the left, there's a preheat oven option,
Oh, yeah.
but it's already heating up right now, so it's disabled.
Okay.
So you don't have to worry about that. But you see the oven temperature is already at 31 degrees, so be ready.
Do you guys recommend preheating, or is it not necessary?
If it's already warmed up, it'll run faster.
How long to preheat?
Maybe like 10 minutes before you start your run. Yeah.
I like that the things you can't select are grayed out,
Yep.
and the things you can are blue.
So you can also turn on the LED to highlight some of the instrument stuff. So
There.
turn on some of the consumables.
Yeah.
Since it's running, you can't really open the door, but there's that CBC buffer right there. Which is familiar with the polymer pouch, and then the ABC buffer. Same array, slightly different from the 3500, but similar format here. So this is a 24 cap array. Yeah.
Okay.
So you open the oven, and you just easily install a new one if you want to.
Okay.
Did you change anything on the consumables as far as like they can only be on the instrument for 2 weeks?
Yeah, so if you go back to the right on the screen, it shows you all of your status of your consumables. So your capillary, how many injections that's left, the polymer, and the installation date and the expiration date, as well as your buffer. And here's your spatial calibration, your remaining injections, and then all the calibrations that were done for your die sets that you can run. And then on this side, you should see some of the maintenance stuff that needs to be done. Some of them, you might need a software update from time to time, but it'll tell you when you need to do all your maintenance.
Okay. And like the wizards kind of for that stuff?
Yeah. Good. Yeah, you want to go to that, you can X out of that.
Sure.
Click on action. Click on maintenance. Yep. And then maintenance wizards. So here you have a list of anything you want to do for maintenance. You can change your array, change your polymer type, change your buffer, change your polymer. And each of them have a video embedded, so you can actually watch some of them for a little bit. So now, like I showed you before, you can actually run multiple plates at a time. So click home. So now it's unlocked, so you can open it up. So
Okay.
you put that in position C. All right, it'll lock again, but now you can link a plate. So create a new plate file. And here you can name your plate. So just click plate name, yep. Type whatever today's date. And,
Okay. Plate 2.
all right, enter. Go to application type, right there. Change it from sequencing to HID. Yeah,
Done?
yeah, done. Go to your plate. And then you can click on the pencil button, click on injection protocol. One more time, yep. And then you can pick which die set you want to run.
Okay.
And then you can change any of the parameters. So this injection protocol pre-populates these fields for you. If you want to do any of the custom ones, you can change them here.
Okay.
So done.
Done. And
All right.
Start? So will it run in the order
Order
that you
set it.
set it?
So this one will run after that first one is done. If you wanted to change it, since it's already started, you can't do that. But you go to run queue, you'll see the order of things that were done.
So I can take plate 2 and go
Right. Right.
top of the list.
Right. So these ones are completed. So plate D is running. So your plate C is going to run afterwards. So it's in queue. And it'll show you the estimated start end time.
Okay. So you have to remember where you loaded it.
Exactly.
Okay.
Yep. So you can click close. And since these are already done, you can actually look at some of the data on there. So you click on the screen, click view sample list. So that's what we ran. This is just the installation run. So you click done, click on action. So you can either unlink, but if you want to look at the data, click on the green icon. And that's what was run on that plate. So you can use this eGUI or the screen to like scroll through, expand that blue bar if you want to.
That's cool.
Oh.
Click on any of those menu, the bullet point buttons on the side. Yep. And then you can select the dyes that you want to see.
Oh.
Yep. So you just turned off blue.
Oh, I'm turning them off.
Yep.
Okay.
Yep. So now you're just looking at the size standard orange. Oh, there you go. And then when you want to bring the samples back in, just click on the color again. Yeah.
Fine.
And on the right side, there's another option where you can zoom in and
Oh, there you go.
and then the middle button
This one?
is your EPT. So it shows you the record of all of your run. There's the oven temperature, your current, your voltage throughout your entire run. So same thing, same touchscreen interface. So, and then when you're done with that, just click done. Click done.
I like that black background. It's so much easier to see the colors than the white.
Yeah.
So let's say you want to run another plate. These all are filled. You can unlink a plate by just pressing B.
Do you want to try?
Okay. Sure.
Unlink the plate. It's going to confirm that you want to do that. So now you can open the drawer,
Great.
remove the plate, and if you have another one ready to go, you can pop it.
Oh, and it shows that it's removed.
It will sense the plate.
Right away.
Yep. So I'll take that.
Okay.
So now you can leave it there, load another plate, or
Awesome.
have it on standby. Yep.
Very cool.
I noticed this is your computer now. You're
Yeah,
not
it is.
hooked up to a computer.
Yep.
So currently, to do like a reboot of the system every 2 weeks, we'll shut down, restart. So you don't need to do that for these?
Yeah. If you want to restart for any reason, you can just power off when no plates are on there, it's not running, and power back on.
Is that something recommended, or can we just leave it on?
You can leave it on, yeah. There's no reason I would need to turn it off, so.
Nice.
It's nice.
Yeah.
We talked a little bit earlier about having the computer and the instrument hooked up to Wi-Fi and being able to control it from your phone and everything. Forensic labs are always concerned about security, so I wanted to ask about that.
Right, so these instruments can be controlled with SAE, so like the security audit and e-signature functions. So if you have it on the network, if anyone logs in, they have to have their own account, log in. If they want to start a run, they have to have permission to sign off. If you don't want to have it on a network, you can have a local computer attached to the instrument, and it's also controlled by SAE. Wi-Fi free, just local area, just within house, so no internet needed.
Yeah. And then as soon as this run is done, we can access it on the computer, any computer with a Wi-Fi,
Right.
on our phone.
Yeah, you can do remote access. I do have it on my phone. You can monitor the run, and I can show you that as well. So from our remote monitoring app, you can remote monitor your run. So here it has a run that's already completed. Click on View Raw Plot, and now, Ryan, you can scroll the data on the phone just like you would on the Flex.
Okay.
You can see all the peaks.
Yep.
Yep. And you can click on different capillaries on top, and then you can filter by color.
And then just
Uncheck.
let's do.
Yep. Apply. There you go. Those your size standards.
Yep.
Scroll and go to all the capillaries. Yep.
Okay.
And it will save your preference on color. So
All right. Nice.
there you go.
So what's coming off the instrument is being directly
Yep.
screened to your phone.
Exactly. Yep.
Like it. Neat.
So we've had a look at the SeqStudio Flex a bit. I know we talked about how the consumables are the same except from the array. So I just wanted to show you really quickly how you can change the array and that sort of process. But you could open it up, these big barn doors. And then if you want to open up the array, I may as well get you to do it, just to show you how easy it is. Open the array door. Unscrew the detection cell. Loosen the cap bundle. There you go. And then loosen the bottom door. It's down here. There you go. And you can slide that out slightly. And then you can just use this little handle here and just pull that out of there. And it will hold it in place. Just remove it.
Great. Where am I?
So the very nice thing about these new arrays are they have, to make it easier to do it one-handed, they lock in place. So you can hold it, and you can install the array one-handed. So it'll stay like this. When you want to load it back into the instrument, just slide it in place like that, and then you can release it. Feed it into the bottom. The detection window is automatically there. It's in the right place. The instrument knows where it is. So you can just tighten everything back up again. Close the doors. Tighten your football, the ferrule at the end, and that's it. That's changing the array.
Easy peasy.
Wow, very easy. The nice thing about this instrument, the very cool thing is, when you change your array, what do you normally have to do after that?
You run a spectral and a spatial.
Right. So you run the spatial because that's how the instrument knows where each of the capillaries are. You run the spectral because that's teaching the instrument the dye set that you're going to be using for that.
Right.
And to make sure it's applied to this new array. The nice thing about this instrument, you only need to run that spectral once.
That's crazy.
Wow.
Like throughout the entire life of the instrument.
Correct. The instrument will remember the spectral you have run. As long as you're running the same dye set for that chemistry, it will continue to apply that every time you change the array.
That's really cool.
It's incredible.
Yeah.
That's amazing.
Algorithm remembers it. It makes sure your data is still there. The only time you'll ever need to run the spectral is if you change dye sets, so a different type of chemistry, or you are troubleshooting.
Wow.
So it can save you a lot of time running this instrument.
Especially, it's one of those things. It's like I feel like we do it, but not enough to really like learn it well every time you do it. So it's, hey, we never have to learn it again.
Exactly.
Break out the manual every time.
Every single time. And the other thing, so obviously, a lot of these things on the instrument, you don't have to do that often. So what we do have on this instrument as well is to help you is called the Learning Center. So if you go to Actions, and then Learning Center, from here, you have helpful videos that you can run that will walk you through when you need to do different steps on the instrument.
Oh.
So if you wanted to learn about how to set up a run on the instrument, and it will take you through all the steps you need to on how to run and set up a run.
Wow, that's cool. For me, I learn a lot better with videos too. Like, hey, here's how you do this, instead of like, reading something that makes it a lot more difficult to be like, oh, take the plate and put it here or do this. It's like, oh, video shows you exactly how to do it.
So now we have everything for visual kinesthetic learners, no matter what you do.
Yep.
This is really great for new analysts also.
New analysts, maybe analysts who haven't run the CE in a while, you're just coming back into the lab. It's just making it so that it's easy, it's approachable, and you just know what you're doing.
Yeah.
So you can always feel confident when you run the SeqStudio Flex.
So back to the, kind of old school almost, like barn doors feels nice, but I also like the familiarity with it too. It has kind of this, you haven't changed everything inside. We don't have to learn an entirely new instrument. It's just almost like an upgrade to the instruments with a whole bunch of cool features.
All right, guys, we've had a really exciting day and an action-packed day learning all about the SeqStudio Flex. I really just want to know what you guys think about it.
It's awesome. I think the design is really sleek. It's user-friendly. You can take your data on the go.
Yes, I love the onboard computer, so you don't have to restart and link these two separate things, and that you can remote in and from home or from your desk, monitor your run, set re-injections, put your plate before your coworkers.
Exactly, yeah, and I even heard you ask some really important questions about data security and stuff while you were there. I mean, that is something that we know is a huge challenge for forensic labs. The world is moving connectivity and cloud-based things, and that's why at Thermo Fisher, we have a group that is dedicated to testing vulnerabilities on our software and our cloud-based systems. And we have teams that are actually there helping you so you can bring it online and work with your IT groups within your lab to figure out what is possible to do within your lab. Because what we're finding is there's a lot of stuff that we can actually do and still be very secure. The old school methods of actually using USBs is not the secure method anymore. It's actually having connectivity with encryptions and secure passwords. So it's going to be really exciting for the future. And this instrument is definitely going to take us there.
I also like the footprint of this instrument. For something that can hold 4 plates and kind of run all of these things, it's about almost the same size as a 3500. So it's taking up a nice small amount of space, As long as it's deep enough at your lab, you can get a couple of these, I think.
Right. And you don't need that control computer either, if you don't need to. So in terms of actual bench real estate, you're a lot less.
Yeah.
And being able to put another plate in the queue while one is running. That feature I absolutely love. I've spent a lot of time waiting for my coworkers to finish up their run before I can even start prepping my plate.
And how do you think that this would benefit you in your lab?
New instruments or kind of new technology comes out like once every 5 years or so, I feel like, and this is just kind of taking us to that next step of, the fact that it has the Wi-Fi capability or the cloud-based capability. It just kind of has that feel of, hey, we are getting into that next generation of instrumentation. So I like it. It's staying current with everybody else's kind of phones and computers and what we're used to, instead of running on some old instruments.
Right, exactly. And if your lab is not ready for that, you can also do it the standard way as well. So it's
true.
Very versatile in that sense, but it's going to be ready for the future when we're there.
Yeah, the fact that you still have the USB, people like to get in their steps still. So it's like, hey, you don't have to use your phone. Go and get those steps in, walk to the instrument again, take it off, and take it back to your desk.
And what do you think about the onboard learning center? Do Do you think that will help you in the lab?
I do, 'cause I know sometimes when you have to like put new polymer on and you haven't had to do that in 8 months, I have to pull out that manual and figure out how to reload my polymer or something. So now I can just press play, see how to do it, pop it on, I don't have to worry about it.
Yeah, the fact that you can actually pause the video too and do that while you're doing it. The doors are separate from the computer screen, so you can just pause it, do that, okay, click play and continue on. It's nice.
And watching the video of it happening in real time is a lot easier than having to read through the wizard like we do now. So that's really nice.
And what about the startup sequence? What do you guys think about that?
I think it's great because currently, like I said earlier, with the computer next to your instrument, you have to turn this off, turn this off, turn this on, wait for a green light, open your software. So now it's just one button, turn the instrument on, and you're ready to go.
And what do you think about the array?
This was absolutely amazing. So can I?
Yeah, please.
So you literally just squeeze that and it releases.
Exactly.
That's really cool. That was something I didn't know that I needed.
Yeah, exactly. No more fumbling around with the detection cell window, trying to get that in place with the magnets and then making sure it's just right. And
Not breaking it.
Not breaking the array. That's key as well. But yeah.
Yeah, I was always worried. I'm like, oh, the capillary needs to be changed. I'm like, maybe I'll just pretend I didn't see it on the instrument. Someone else will change it, because it's like this thing is like, you know, kind of all over the place. So it's nice that everything's kind of in place. You know, a couple clicks, couple knobs to turn, and it's in and ready to go.
Yeah, you can do it one-handed. No more 3 hands and tape needed. I know we've had an action-packed day. It has been super exciting from our side. Thermo Fisher Scientific has loved having you here, so thank you so much for taking the time. It has been great. I really hope you got a lot out of it, and I can't wait until the next video.
Thanks so much for having us. This is wonderful.
I had a blast.
You were the first accredited forensic scientist to do a run on the HID version of the SeqStudio Flex. This is great. Thank you guys so much.
Thank you.
Thank you.